Okay, so today we're going to be going over the Los Alamos National Labs database. This is a very important, so I'll edit this a little bit later. So today we're going to be going over the Los Alamos National Labs HIV sequence database. You can find this by Googling LANL and HIV or HIV sequence database. I may, nope, I don't have it yet. Just go here. And so what I wanted to do was just kind of give a very brief overview and emphasize some of the areas to get more specific HIV resources that we'll be using in the near future. But I also wanted to use this as a brief opportunity to talk about HIV bioinformatics in general. So what we're going to be doing, this is the main page for the LANL HIV databases. We're going to be going to the sequence database. And so just kind of going through what we see here, make it a little bit bigger. You have your main menus on the top part of the screen. You have a couple of other ways of looking at some of these resources. And then on the bottom here, you'll have any updates that may be relevant. So if they change any of the tools, things like that. And so what we can do is just kind of start going through the top part. So we have the sequence database up here, which is where we are, and then these are other databases that we won't be getting into today. We have a search interface, which is some of what we see down in the programs and tools. Search database is the best way of getting very specific sequences, and we can just jump in there briefly. And so this is really one of your power tools to be able to get very specific types of information that are organized. Here we have different sections. So we have sequence information, and this sequence information is using metadata that is tied to individual records. And then you also have a little bit more detail that you can ask for as well, if you already have a sequence ID or an isolate name, for example. And then different sequences have different types of metadata associated with them, and we can talk about that in the near future. You can also look at different parts of the virus, which we'll get into briefly in a little bit. You have other types of ways of looking at these sequences. For example, you can use nucleic acid information. You can also use protein information. That's what this search box is asking for. If you have a publication that is from a work that had deposited sequences into either NCBI that are indexed in this resource, you can use that publication ID. Some of the metadata associated with some of these sequences also includes patient information, like if it was from a clinical study, you can use that as well. Some of the data includes some information about where the sequencers were obtained, usually, for example, which country they're coming from, and in some cases, city and some other geographic information. You can look for amino acid motifs as well, and then you can also specify how many results you would like to go through. So just briefly, we can go through one of these examples. Let's say I want to look for all HIV-1 sequences. So, and of the types of HIV I can look for, the types of related sequences I can look for HIV type 1, HIV type 2, SIV, which is simian immunodeficiency virus as opposed to human immunodeficiency virus. And just commenting briefly, so HIV 1 and 2, they're roughly related, but we can treat them as two different viruses. They're clinical, they infect somewhat overlapping cell types, and then HIV-2 winds up having a more indolent clinical course. folks usually don't get as sick as quickly or as immunodepressed as quickly, but they really do have two different types of origins. So it wouldn't necessarily be um, appropriate to like lump all HIV types as one type or two types. So most HIV work that is done, and most folks that are infected with HIV are in fact infected with HIV-1. So we'll be sure to emphasize those points. SIV is a simian immunodeficiency virus. It generally is not floating around in people. And SHIV is a hybrid between simian immunodeficiency virus and human immunodeficiency virus. These are usually more relevant for sequence type comparisons, not so much clinical comparisons. And then we also have synthetic DNA, which is like, let's say, if we're making mutants and things like that. But synthetic DNA is not found in people. It's not found out in the wild. And then null just happens to be if the sequence was deposited and the depositor didn't necessarily specify whether or not it was any of the above. So let's focus on HIV-1 in this case. Subtype information is useful sometimes because out in I don't want to say the wild, but out in the real world, whenever patients are getting infected, there are slight differences in the HIV sequences, and this lets you actually group the many different types of HIV-1 into subtypes. So most HIV strains in the US are actually B clades, so we can just focus on HIV-1 and then B subtypes, I'm sorry, and these are all usually in the major clade. You can see here. You also have what are called circulating recombinant forms. These are the CRFs, and these are recombinants of the different subtypes that you'll see after this underscore. So in this case, CRF01 is recombinant between A and E, and there's a resource in the Los Alamos National Labs HIV database that actually explains this in much more detail, and I can go over that in a separate chat. So let's focus on B subtype, make sure that's selected. We're going to leave all of this other stuff open. I'll talk about the co-receptor the specification in a later video. Just going through the next field, I want to focus on sequences that have complete genomes. So I'm going to select complete genomes, and then this is going to greatly narrow down the amounts of hits that I get to my query search. If I wanted to have a much more comprehensive view at a particular region of HIV, I could, for example, focus on the 5' LTR, which would just give me sequences that would have that would happen to cover the 5' LTR. If I wanted to focus on the, let's say, the protease region along the pole gene, I could do that as well, and it would return sequences that that have at least the protease. So the idea is that you can focus on which parts of the virus you care about. If you care about the entire virus, you would select complete genome. If you care about specific parts, you can pick specific parts. Yeah, so just scrolling through some other things. I think we can leave that by itself, so we can just click search. And again, we're going to be looking for HIV-1 sequences from B subclade or subtype and hits that only have full-length virus. So just going through what the results look like, it'll tell you how many sequences are being shown. So 1 out of 100, or 1 to 100 out of 5,703 sequences were found. And again, these are in all of the HIV sequences that are indexed in this source, which is generally drawing from NCBI indexed sequences. We have of the sequences that match to HIV-1 and B subclade or subtype and complete genome, we have this 5,703. It also notes how many problematic sequences are removed from this final output of 5,703, and that kind of bears a different discussion. So as kind of a homework exercise, I would encourage y'all to actually look to see what this resource is calling problematic sequences. But it's good that it does this just because it gets rid of some redundant sequences or sequences that may not necessarily be that useful for downstream analyses. So just going through kind of what the different columns are, this output allows you to select different specific sequences as you're looking through them. Because there are so many, that may not necessarily be useful for these applications, but it allows you to select them, and then what you can do is actually look at the sequences later. What you can do then is you can actually blast that sequence against other types of sequences, and that'll basically be using like an NCBI blast type situation. And this requires another discussion for sure. What you also have is, in some cases, you have patient IDs, or in other cases, you have the names of the different types of viruses. So this LAI, this is a very, very early isolate, one of the first isolates, if I'm not mistaken. I think it is the first isolate. And we can look at that in a little bit later. And then you also have different accession numbers. These accession numbers are useful because they will allow you to pull up those records in the future, so you don't have to include all of this information in your search queries. You can just use these accession numbers. Each of these sequences has a name. In later studies, usually the name of the sequence will be tied to a patient ID code. This patient ID code is something that other people can use to keep track of all of the information without necessarily violating a patient's information rights or privacy rights. Here we see what the subtype is. So again, this is a good positive control, or not a positive control, this is a good quality control for the query that we ran in this database search engine. So we wanted all B subtype, and we got all B subtype in our returns. And we would actually have to verify through all of the 5,700 and what have you, just to make sure. But we're trusting this database to do its job. Yeah, so, and I can say that just because I'm familiar with how it works. What else? We have country. So for example, the first sequences were in fact isolated from France. And then we have sampling year as well. Note that the sampling year isn't in order. So that's an important feature. What else? What else? We do have, and if you clicked sampling year, you can click it and then it will reorder. And this is just kind of a common search feature. We'll let it do its thinking. Or not. Let's save some time. So what else do we have? We also have these little cartoons, which we'll look at briefly next, so that this makes a little bit more sense. But these little cartoons are actually representative of the genes that are in, and some of the features that are in HIV-1. And we can see here that there are two colors. There's green and there's yellow. So green is the interval of the feature, starting from the beginning of the virus on the left side to the end of the virus on the right side. And this yellow starting point, you can see here, captures about the second half of this first green block, which is actually the 5' LTR. It scans through the rest of the virus, and then it almost finishes at the end of the last green block, which is the 3' LTR. And you can see, if you look very closely, that these different viruses have different amounts of this coverage, which is the span of the sequence that was deposited. And so these green blocks are with respect to an HIV reference. In most cases, it's HXV2, which is actually this first sequence here. And so it just goes to show that different sequences, if you look at them, will have different amounts of the virus for some reasons that we can get into a little bit later. You also have the sequence length here. So in some of these cases, you don't have full length, or you don't necessarily have the same length of all of these sequences, which just again goes to show that you have different amounts of virus for slightly different reasons. In general, most of these sequences will have a pretty uniform amount or a pretty complete amount of the 3', and then different sequences will report different amounts of 5'. We'll talk about that. And then another quality control step for quote-unquote organism, we see here that most of these sequences, or all of these sequences, I'm sorry, are HIV-1, and that's what we want to see because we, again, asked the search engine in the database to return these values. So let's say we want to look at this first sequence. So we click that first, we click that first value and then we hit next. And it should return us that single value. It may need to think a little bit more. Oh no, I'm sorry. So what this next and last does is it goes through the different queries. So on the first page it was 1 through 100, and the second page is going to be 101 to so on and so forth. So what we can do is, what I was trying to do, I'm sorry, is click this, they do have some earlier sampling years. We can look for this one that was selected here, which was the first one just by default. And what we can do is, let's say we want to look at a few, so we want to use that one. That one is the reference for HIV-1. Let's look at these other early HIV sequences. So most of them are from New York, and you can see that they're NYC, and then one of them, or a couple of them, are from San Francisco. So let's make a tree with those just to see kind of how they're related. And then so this HXB2 is actually very similar to the LA sequence. Like they're very, very closely related. And then we don't, and we can include subtype reference sequences. And what I predict this tree will look like is that there are different subtypes, so A through some of the recombinants. And so most of what we're looking at should be clustering in B, and then there should be a slightly different clustering pattern between the San Francisco sequences and the New York sequences. And then this LAI sequence from France should be very similar to HXB2. So we can click OK. We can see what its output looks like. And then we can just use the default. We just use the default sequences here. And we can give it our email. So let's say just say test tree, and that may take a little bit of time, or it may spit us out some results. Okay. So this is what our tree looks like. And so most of these are actually the subtypes that we were looking at. So remember that we were pulling mostly Bs. So all of our search criteria, or most of our search criteria, are actually down here, and I can talk to you about what the different names mean. But these above are different subtype examples, and so these would actually be useful if we were looking at other subtypes. For example, if we were looking at examples from subtype F2, they would be around here, and these would be our control samples for F2, just to make sure that we have something nearby to compare to. Because if you're just lumping a bunch of sequences that aren't necessarily closely related together, the tree type or the dendrogram diagrams that you get, or the relationships that may seem to occur, may not actually be real or useful to look at. So what we're going to do is focus on this part here, and we can see that we have this outgroup, which is an F2 in this case. In this case, this is the most different sequence compared to all of the ones, or all of the rest of the ones in this dendrogram, or in this tree. And so what we can go down here is go in and look at the different branch points. And so the length of these differences is actually important for different types of analyses, which require a different video to go over. But we can see here that there are different branch points, major ones between F2. So these are all kind of together, but this is the most different compared to all of these other ones. What else? As we go in, then you have everything else, and then F1 breaks off, K breaks off, J breaks off. And then between K, you have these other ones. So in there, J breaks off. J breaks off between G and A, and then A kind of clusters over here. Between that, if we go up a little bit, we have H and we have C. And then going back up a little bit, we have D and B seem to be pretty close together, but then B winds up clustering. And so we can see here, the way the names are broken down is the subtype is the first value before a period. The country, which is a two-letter code, is in the second space between the first period and second period. So most of these are in US, except for the ones from France. And then we have a couple from, I think TH is Thailand, I want to say, and NL maybe the Netherlands. I need to double check that. Then we have the year that the isolate was made. So remember that we had a lot of these older isolates from the US that are up here. We also have, let's see. So these Thailand and Netherlands examples, remember, we actually didn't explicitly ask for them, except during the stage whenever we asked for references from different subtypes. So this Thailand and Netherlands, these are just the references that they added here. In addition, the HXB2 is the overall reference for HIV. And so remember that the Li3 and this other one, like, they're supposed to be very, very close to each other, but they are different because the HXB2 actually has a little bit more sequence upstream and downstream. This other isolate, I forget whether or not there are internal differences, but I know for sure there is a difference between the ends and the total sequence length of these. What else? And so we do see that the New York viruses tend to segregate away, or they may have their own clusters, but they do tend to separate away from the San Francisco viruses. I think a better way of explaining that, though, is that the San Francisco viruses segregate away from the New York viruses, although that's not necessarily the case, because it does seem like there's some relationship between some of the New York viruses and the French viruses, and that kind of makes sense in hindsight, just because New York is a big global hub. I'm sorry, not French, San Francisco. New York is a big global hub, and San Francisco is a big global hub at the time. So, yeah, so that was what I was trying to say. Yeah. So, and this is a pretty cool thing to look at a global level. And then what you can do also is you can look at smaller parts of the virus from different areas and in different conditions, and you can do these types of analyses to see, in general, at a zoomed out level, are there maybe relationships between sequences or sequence homology between several different classes of viruses, and then that allows you to think about ways in which those homologies may have occurred. So there may be functional reasons why there's homology, or there may be evolutionary reasons for that homology. So this is pretty cool. We can go back to the HIV sequence database. So we were talking about the searching, but what we can also do is just kind of go back to where we were before we started the search. And if you go here to Program and Tools, the search interface, this is another way to get to where we just were. So again, it gives you the ability to start over and look for whatever sequences you want to look for. There's also advanced search, which allows you to, in a much more streamlined version, basically pick and choose which pieces of information you want to look for. And this is really useful whenever you have a bioinformatics approach to get this information, instead of actually going through and using this search interface to select what you want. Moving away from just generally pulling whatever sequences are available, you can also look at the alignments that are made. So to understand the alignments that are made, it's actually, I think, useful to look at the compendium downloads. And so these compendium downloads are basically summaries that were made roughly about every year to describe advances or additions to this general resource, the HIV sequence compendium. The compendium is essentially a very good introduction or a timely summary of the information that's contained in the sequence database. So we can look at the first one, and we can look at the complete compendium just briefly. And so this has a lot of information, and it's really, really good information. I would say it's one of the key resources for HIV bioinformatics information. Just skimming what's in this resource, and I encourage everybody to read it. At least the latest edition. You have just kind of a general introduction, like, okay, like what is the compendium? If you're going to cite it, how do they prefer you to cite it? They give you genome maps for HIV, which are very important, and then they tell you a little bit about what the different proteins are and a little bit about the different landmarks across the genome. For example, non-protein coding regions and gene regulation regions. And then they give you a brief explanation, and then they jump into the different types of sequences that are in here. And so this is, again, very important. And if you don't read the beginning, you won't necessarily understand the output. So you just skim through real quick. This similar information is available in the HIV database website by itself, but this is a much more, I think, useful way of looking at this information, because it also puts it in the context of some other very closely related retroviruses. So, for example, we have HIV-1, whose reference genome is HXV2, and this is just one of the first HIV sequences to be publicly recognized. There were some other sequences, remember, that we saw from the 70s, but those weren't necessarily available for early studies. So HXV2 was the first one that people have been using up until very recently. And so we have the HIV-2 reference genome, which is BEN or BEN, however you want to say it. And you can immediately see that these two viruses look pretty close, but there are some interesting differences. For example, the polymerase genes have different lengths. There's a little bit of a difference between the packaging region, or the psi element, between HIV-1 and 2. HIV-2 also has this VPX protein. HIV has VPU. We have glycoprotein or GP120 and 42, which together make a GP160. We have these together, and then some of these other proteins have slightly different lengths because they do slightly different things in different viruses. Also note that HIV-2 is a little bit bigger than HIV-1. We also have SIV here, which the reference genome for that is Sykes. And we see here that these viruses are also pretty similar to HIV-1 and 2, except for there is no VPX and there is no VPU. And then there is a slightly different organization scheme for these viruses as well. But most of the parts are there, and it's believed that most of them behave pretty similarly. Going through, we have a table that talks about specifically what each part does. And these are just very brief summaries, so I encourage folks to, if they're interested in specific proteins, to actually take a deep dive and look to see everything that's known. For example, we care about HIV renal disease in our lab, and VPR and NEF are really important pathological proteins. They do bad things to the kidney if they get inside. But note that the information that's reported here is usually with respect to these proteins' effects on immune cells. So NEF, for example, is a CD4 and class 1 downregulation factor. And it also has a post-translational modification. Sometimes the protein gets myristoylated. VPR promotes nuclear localization of the pre-integration complex. It can inhibit cellular division, and it can arrest infected cells at G2 and M phase. And we do see that in some renal cells, but this is still kind of an active area of development. Whether or not this is seen in all HIV-infected cells is still kind of being studied. We see some other landmarks of the HIV genome, including the genes. Briefly, we can go back up here to these gene maps. Whenever I say an HIV gene, what I mean is GAG, for example, or POL, or envelope, or sometimes VIF or NEF. These are specific regions that may code for multiple proteins. And so, very importantly, these HIV genes that code for multiple proteins, the specific proteins that are being coded for in these genes are actually denoted by the internal nomenclature. So, like P17 is a polyprotein that gets cleaved off of. It is a protein that gets cleaved off of the polyprotein that is what GAG winds up getting translated as. Whenever we have HIV mRNA that can support the translation of that information. And that gets very, very complicated, but I did just want to emphasize that point that GAG isn't necessarily a functional protein by itself. POL isn't necessarily a functional protein by itself. These are polyproteins that get cleaved into their respective enzymatic activities. Just skimming down a little bit more. They give you kind of the starting material, like these are amino acid codes and these are nucleic acid codes. And then once you get into the complete genomes, they give you a rough map as to where things start and stop with respect to the reference genomes that we were just talking about. HXB2 for HIV1, BEN for HIV2, and CYKS for SIV. And then after describing where different features are, according to HIV in this case, they get into a list of the genomes that are included here. And then just briefly, the full names of the genomes, and then the abbreviated names are towards the end. And we talked about this nomenclature, subtype, country code, year that it was isolated, and so the last 2 digits of those years. And then the abbreviated name for the sequence, in this case HXV2. We have the accession number, and if you click this, it will take you to NCBI so that you can look at the accession number. Let me just double check that to make sure, because I haven't done this in a while. It may also take you to an internal version of this. Yeah, okay, so it takes you to an internal version of this. And this is, again, just very similar to what we were looking at before, except this Li3b, this is the full length version, and you can see, confirm that here. And then the full length is 9,719. But we can go back to what we were looking at, which was the compendium. Oops. Just re-download that and zoom down a little bit. What else? Country, author for that study, and then this is the reference. If you're going to be using the sequence, you can either cite the accession number or you can cite the reference, depending on what your preferences are. It's always nice to cite the reference. But the accession number nowadays is almost as, so the accession number is more useful than the reference, because usually the reference you have to kind of find where the information is. But either the accession number or the full reference are important ways of citing using this sequence for downstream applications. So just skimming through, these are the sequences that are in this summary, and then we can actually look at what the compendium is. An important part of the compendium is a sequence alignment, and not any kind of sequence alignment, it's a multi-sequence alignment. So you can see these are, for example, the main HIV-1 reference, and then you have a bunch of A1, A2, A6, and then going into the Bs, a lot of these were ones that we were looking at previously, or some of these are ones that we were looking at previously. So we have that TH, and then I don't think we have another one in here, but TH is one that was in here. We have Cs, we have Ds, we have F1s, we have F2s, and so on and so forth. Remember that the 5' LTR is the beginning of HIV, so the very, very beginning of the reference strain for HIV starts out with T, GG, A, GG, and so on and so forth. What a multi-sequence alignment is you're taking, and there's the assumption that you have related sequences, or sequences that may have some type of homology, but you're taking multiple sequences and you're asking a computer program to fit or to basically say like where certain sequences align to other sequences. So the way that this one was made is you are aligning all of these other sequences to this reference sequence for HIV, which is the HXV2. And so it tries to do that in the best way possible. And so there are a couple of different things that I have to go back and look at. But you have dots and you have dashes, so we can actually look to see what those mean. Over this table, they should be able to tell you that. Yeah, so for the nucleic acid codes, we have the normal ones, ACGT, U, if the sequence deposited a U, which is a little bit rare for this type of resource, but it can happen. And then you also have these combination nucleic acid codes, M, if you wanted to say this could either be an A or a C, and they're not sure about it, versus the other things. These are another way of calling them as ambiguity codes. And then N is there is evidence that there's a nucleic acid there or a base there, but you don't necessarily know what that base is. And so what we care about to understand what we were just looking at is we have these dots, which in this resource code for gaps, and then we have these dashes that code for identity. So depending on the resource, sometimes a dash is a gap, so it's really important that you look for this type of information in each resource, or in each new resource that you're looking at, just to make sure that you're understanding what you're looking at. So, and it had been a while since I looked at this resource, so I needed that refresher. So dashes are identity, dots are gaps. So anywhere we see a dot, it means that there wasn't coverage in that area. So remember those sequences that didn't have full length yellow coverage over the 5' LTR? So these are sequences that if we looked back at their cartoons, it would look like that. And this is actually very, very common to not have coverage at the 5' LTR. And one of the reasons why is because a lot of the sequencing technology that's used to make these sequences don't actually cover this part of the virus that has a very special functional consequence to how the virus is made. Usually with these resources, there is very strong 3' bias, so you're picking up the end of the virus first. And this 3' bias is much more apparent with newer technologies. Older technologies, actually, we're looking at the proviral sequences and trying to clone proviral sequences, and usually you had a higher likelihood of picking up the 5' and 3' LCRs. Not always, but that was a common feature of older HIV genomes. And we can talk about that in the near future. The dashes are identity. So for A1, everything's the same. So this is actually also a T, this is a G, this is a G, this is an A. And then our first difference is actually at this fifth nucleotide, where instead of an A, which is what we see in HXV2, we have a T. So, and then so on and so forth, 3 bases that are the same, and then instead of a C, we have a T, and so on and so forth. You can start to skim through many different sequences, and so where there are gaps or where you can't see what is actually in the sequence because that wasn't captured by the sequencing experiment or by the depositor's experiment, you can start to see that those differences may actually be conserved in other viruses that are loosely related. So for the A's, it seems like of the A's that are there, all of them have a T in this area, except for in the sequences that you can't see what's going on. And you can actually see that most of the sequences here are actually fairly conserved, which is what we would see whenever we're looking at different viruses that we're claiming are related to each other. So this is pretty sparse, so we can jump into areas that have more coverage. And we can start to see here, there's some other things that we haven't talked about yet, where they are adding those features in the tables that we were looking at previously, but on top of HXV2. So we have things here, for example, let's go down a little bit more. Let's say down to we have some areas that are conserved. Remember, we have all of these dashes. These dashes are pretty important, and anytime you see a letter with this nomenclature, this letter is different than the reference. So many of these different viruses, like these recombinants, have A's, for example, at this position instead of a G. Some of them have T's here. And so you start to see like what differences are tolerated by these viruses versus what differences aren't tolerated. If you don't see a base there, it may be that the sequence can't support that because all of these conserved regions generally have a very specific function. And then these are all non-structural components. This is part of the packaging structure. And then immediately after the packaging structure, or a little bit after the packaging structure, you start to have protein coding sequences, and in general, these protein coding sequences are fairly conserved, but you do see some differences like throughout here. So all of these are protein coding areas, and then you do have some slight variances. Most of these are actually synonymous mutations, where they may not necessarily be protein coding, or if they are protein coding, they may be conservative mutations, where the difference between switching out one type of amino acid for another type of amino acid may not be that important depending on what the amino acid is. So this is skipping over a lot of basic biology, or a lot of college level, entry level biology, to understand the types of information that go from DNA to RNA to protein. But understanding those concepts will be useful for understanding the type of information that can be seen in the compendium. And then also that type of information is useful for being able to understand what types of questions you can ask with these sequences. So we were talking about just generally pulling up HIV sequences. We hadn't talked about the curated alignments. So what we were actually looking at were examples of the curation process, where we're selecting particular examples of different subtypes, and then we're doing alignments back to HXV2, which is the reference for HIV1, and that just allows us to have a starting point to compare other sequences to. So what we can do is go back into the curated alignments. You have different types of curated alignments. You have the compendium alignments that we were just looking at. You also have web alignments, which have slightly different meanings, and you can click in each of these names to figure out like what the actual difference is. And this will give you different starting points that actually make sense, because there are certain types of redundancies that are included in pulling up these types of sequences with kind of an ignorant way of pulling up sequences versus a more nuanced or curated way. So depending on what your level of expertise is, you may actually be able to jump in here and get the sequences that you need for what you're using them for, or you may want to start out with these curated alignments. So there's reasons to do either, and so I'll try to introduce you all to those slowly. Next, we have a big section here, which are different tools. These tools, I would encourage everybody to actually just go through and explore. They do different things, and some of what they do is a little bit complicated, but some of those sequence manipulation tools can be useful if you want to compare sequences. And then what else? Some of the alignment tools also can be useful. Some of the tree making tools can be useful because they have a lot of the entry-level bioinformatics that you would want to do with these sequences already established and already kind of validated that they work in here. So that's nice. We talked about compendium publications. We also have some review articles here. These are useful to just help you get caught up with some of the more popular concepts in HIV research. So, for example, the cytotoxic lymphocyte epitopes. So these are epitopes that you would want to try to be able to express so that your body can eliminate cells expressing those. You also have certain HLA or MHC or human MHC basically immune immune genes, which are important in immune response. There was some work studying that and its relation to the progression of HIV, and you have some other older reviews as well. So these are actually pretty useful. Not immediately important for what we're going to be doing this summer, but it's useful if you care about what we have learned about HIV and to understand some of the newer tools. It's useful to understand what some of the older tools are. Here we have guides in our last little tab here, and these are really, really, really useful to understand a lot of the resources that are embedded in this very important collection of resources. So I would encourage everybody that on your own time that you actually go through these guides as well. This may actually be a pretty good starting point, which is kind of ironic because we're ending with it today. But I would encourage everybody to kind of go through here. The CRF guide is really useful for understanding what the different CRFs are. You also have the HIV gene map, which I think is also a good ending point because a lot of you have seen this before in our previous talks, but now you can kind of understand where else this information is stored in this HIV sequence database. Hint, hint, it's in the compendium. And then this gives you a starting point for studying different gene features of HIV. So with that, I think I will stop today, except to be able to continue our next video, which will be actually using a tool called Galaxy, or a set of, let's say an environment called Galaxy, which is a set of tools that are available online where you can submit bioinformatics jobs to online servers, have it work on the background. You can check it on your phone if you need to, and then check back whenever the results are ready and you can continue your work. But what I would like everybody to be able to do is to be able to pull up the reference sequence for HIV. So what we can do is just from this page, we can get our sequence for HXV2 by clicking here. And then this is a way of looking at the GenBank file. This is one way of getting these. Another way is actually going into GenBank and pulling it up. What I would like folks to do is actually to do that instead. So instead of pulling it from here, to pull it from GenBank. So what we'll do is we will take this accession number and we will copy it. Not copying the hyperlink, but just copying the accession number. Open a new tab, type NCBI. So we're just going to be looking through all databases, and we're going to be adding that accession number, hitting enter, and then NCBI has a new output. And so our top hits are going to be added here, and then other ways of looking at these hits are down here. For example, if it's in literature, if it's in different types of gene databases, if it's in proteins, if it's in genomes. Note here that there's one genome, or one nucleotide sequence, and a few SRA entries. What we're going to be doing is we can do a little bit of data exploration on this page. So let's click our first hit, and then we'll see that some of those other links will take us to the same page. So this is the main GenBank entry through NCBI. Just a fancy way of looking at a relatively dry file type, which is the GenBank file. But this has a lot of information. So it has the name of the sequence. It also has the GenBank accession number. So remember previously we looked, just to kind of copy and paste, we looked for this accession. The full GenBank accession is that same information, but it's a period and a 1. and that just shows that this is the first edition and that this final edition hasn't changed. It tells you the locus definition, HIV, HXV2, complete genome or CG. It gives you the length, it gives you the type of molecule, so this was a single-stranded RNA molecule. It tells you that it's linear instead of circular, and it also tells you the date that this was deposited, so this was deposited October 21st in 2002. And so this is a bit of an edited version of one of the original HIV sequences, but it tells you, it gives you kind of the rest of the information, including papers that this came from, some other articles that cited this. What else? And then it gives you the different parts. So it tells you the proteins that are encoded, or the genes that are encoded sometimes. And the kind of tricky thing is that each HIV genome that is deposited in GenBank has a different type of nomenclature, which is kind of annoying. But sometimes they're more consistent than others, but they'll give you the protein coding genes, or they'll give you, by gene, I'll say like GAG or POL, or they may break it down into individual proteins like P17 or P24, things like that. And then towards the end of the GenBank files, they'll give you the origin or the original file. And in this case, remember that it said single-stranded RNA up here. So 9,719 bases that are single-stranded RNA. So in this particular record, they actually did you kind of a favor, and depending on what your application is, it may not necessarily be a favor. They wound up converting that uracil, which would be what you would see in RNA, into a T. And this was actually a very, very common practice, just understanding that, yes, this is supposed to be coding for RNA, but whenever we're using this for different applications, it was just common to convert this to back to a T, which is the DNA sequence. And so the coding DNA sequence. And then we have this double slash, which is the end of the file, which is useful if you're trying to search for what is in the entire file. So what we're going to do, or what we're going to need to do, is we're going to download this in a couple of different ways. So we're going to send to, and we're going to select complete records, and we're going to send to file. And we're going to download this in a couple of different ways. So I want y'all to select here the full GenBank file. So we're going to create that. I'm just going to export that. We're going to go back to File, and instead of full, we're going to select FASTA. So in this case, this is just going to be this last DNA, sequence information that is supposed to be coding for the coding strand in DNA, or the reverse transcribed single-stranded RNA molecule. So we're going to select that. This is going to be in FASTA. And then we're going to get a couple of other types of files. So we're going to get GFF3, which is a format that will allow us to look at different features in different contexts. So we'll just select that, and we'll select get that. And so we're going to look at our downloads file. Oops. We're going to note that we have some things that are just trash, like early practice videos that I don't need. What we have here is just a kind of very generic name, sequence.fasta, or sequence.gb for GenBank, or sequence.gff3.text, which is annoying. So what I like to do is I like to immediately, until I have, or before I have a bunch of these, as sequence -57, and then I have 56 other sequences that I need to contend with. Don't do that. What I like to do is I like to keep the accession number as the beginning. Oops. Go away. What I like to do is keep the accession number, just click it once, and just copy that down, and then underscore, and then something in human readable language that will allow me to look at this. And just collect this first little bit here. and we can add HXP2 actually, because sometimes spaces aren't good for bioinformatics applications. And so what I'll do is I'll just keep that entire string. And sometimes those periods can be a problem as well. So let's just take this string. So the base accession number KO or K03455 underscore HXP2. And then we can rename our GenBank file that. And then we'll have to do a couple of things for this here. Get rid of that beginning part. And then for looking at the information in this file, keeping it as a .text is useful. But for using it in some informatics applications, they actually want to see this ending, this file type ending. So sometimes we may actually need to delete this, but we can keep this like this just to do some exploration. I did want to take this time before we finish today to actually go through these different file types. I may actually separate this as a separate video. We'll see how that goes. But with the idea that, remember, GenBank, actually, I just want to look at the plain text version of this. GenBank, we were looking at it in almost like an XML or like a web page format previously. The actual information that is in that file is not that complicated. Where we have things that are tab delimited, I think they're tab delimited, or space delimited. We'll see. Maybe space delimited. But anyways, it's a columnar type of file where you have these rows. For example, locus here, or locus definition, has certain kinds of information. Actually, no, locus has these fields here. So this is the name actually that this sequence has, or an abbreviated name, and then it has some information about the general information about the file. Definition, it has a more complete name. So this is like, let's say, a full legal name, and this may be like a nickname, let's say. That would be a good way of looking at it. An accession number is a very easy way of getting this information if you wanted to search for it in NCBI. So again, the base accession is this number, and then this is the current version. So in this case, 0.1 is the first version. And then we have some keywords of things that are in this record. So we have tar, tar protein. That's actually not a protein. Well, that would be TAT, I think. Yeah. What else? GAG protein. We don't really have GAG protein. It winds up being included as a polyprotein. But these are other key terms that other people used, or that might be useful to pull up this type of information. Source organism, HIV1, so on and so forth. So we had seen all of this before in a slightly different way of looking at it, and then we end with our fastest sequence in this kind of interesting output. So it winds up being, I think, broken up into 10 bases. So yeah, so this is 5 bases, this is 10 bases, and then you have a little bit of a break, and then so on. So the first 60 nucleotides, 10, 20, 30, 40, 50, 60 are in the first row. The next 60, so on and so forth, so on and so forth, up until 9,719, or 9,717. I always get these confused. No, 9,719. Okay. And then the end of the GenBank file is this kind of double slash symbol, /. And so this is the end of a GenBank file. What we can do is we can also use a text editor to open up a FASTA file. Oops. So we can just select that and open with the same text edit program, or if you're using Windows PC, there may be another text editor that you prefer. This is a FASTA file, or a .FASTA file. Sometimes it's also abbreviated .FA. These files just have sequence information, and it's either nucleic acid, either DNA or RNA, or it may have amino acid information. So we're looking at this HIV genome. This is also HXV2. We're looking at the nucleic acid information, and remember that they did the job of converting the U's to T's for this case. What a FASTA file does is it explains or it summarizes. Actually, no, it doesn't explain or it doesn't summarize. GenBank files did a better job of explaining and summarizing a lot of information, the same information that we're looking at. What a FASTA file does is it just collects that information and keeps it in an organized way so that you can, for example, look through the nucleic acid sequence in HIV, or you can compare the nucleic acid sequence in HXV2 to other sequences, for example, other ones that we were looking at. The anatomy of a FASTA file includes the header line, which has this kind of greater than caret. Usually there's the accession number followed by a few other types of information. So remember, this is kind of like the full legal name. This is just another way of naming this type of information. We chose a different way of naming it compared to this, in part because this is kind of long and annoying to deal with. This is nice and short, and we can actually use this to compare a bunch of other sequences quickly without having to change our windows or anything like that. And so the next line immediately is the first sequence, and there are many different types of FASTA formats. Like sometimes you can just have the full length of the sequence in one line, which is annoying to look at. This is just a very easy way of looking at it. This is either the first 60 or the first 60 or the first 50. I forget how long these are. But these are relatively succinct ways of summarizing the information, and there are even more compact ways of being able to, for example, compress this type of information to serve different needs, and that becomes much more relevant whenever we're dealing with organisms that have much longer genomes. For example, if the HIV genome is just under 10 kilobases long, or like 10,000 bases long, the human genome is something like 3 billion bases, and that's only if you kind of average the information between mom and dad. So if you are considering the information in mom and dad, and not taking into account the different differences between different cell types in the body, a mom and dad copy of somebody's genome will wind up being 6 gigabases, so 2 times 3. So this is how information can be summarized or presented in a certain way as a FASTA file. And then we can also look at the same type of information as a GFF3 file. And so this is going to be kind of an abbreviated way of looking at a GenBank file where we have some headers or just kind of notes that usually aren't read by bioinformatics tools. This is just for people. So we can see that in this case, we're looking at sequence regions, and this is going to be from the succession number, and we're going to have a beginning and an end. So this line is actually very important because this for GFF3, sometimes computer programs actually look at this, and it's important to know when the sequences start and stop. And then it'll tell you where this GFF3 file was generated, which is useful. We're using our chromosome name or number here, or DNA name or number here, or sequence name, I guess, in this case. And so we have that as our first column. We have the format of the file, which is GenBank. We have different classes, which wind up being, for example, just the full length is this first row, so region from start to end. And then we have different breaks. So for example, we have the long-term repeat or LTR, we have a repeat, we have mRNA sequences, we have exons, we have CDS or coding sequences, and so on and so forth. We have some introns that get spliced out. And these are all important descriptors to a gene browser or a program that can be used to look at this information, which will actually use this in a future video to be able to see different features of HIV, to make some cartoons that look like, or to make some graphics that look like some of the things that we were looking at in the HIV sequence database. So the HIV map. This information is one way of getting to that type of pictorial description. We have start and stop points relative to different features in HIV. These are analogous to what we would be seeing in the compendium, except each sequence winds up having its own slight variations to those. We have a couple of other distinctions. For example, these plus sequences denote that these features are all in the forward orientation. And then we also have, from what I remember, we have 1 through, from what I remember, this is 1 through, so 0, 1, and 2, these are different reading frames that are important based off of where proteins start and stop. And then you have a few other bits of information to help you actually give names to different pieces whenever you're trying to display them. But this is, in general, a tabular way of looking at information that we had been looking at previously in other types of formats. So the jargon can get complicated, and part of that jargon winds up being these formats, but these formats all have very particular types of uses that we'll be getting to very, very soon. So with that, thank you for your time, and I'll see you next time.